If one were to cross two flies, one with the wild-type condition of brick red eyes and a second with a darker brown sepia eye, the F1 generation could be examined to determine which of these alleles is dominant. So if these flies are true breeding for their traits, then if the sepia allele is dominant, then all of the flies of the F1 generation would be heterozygous, and all of them would have the condition of being sepia, having dark brown eyes, if this trait is dominant. If the F1 generation is crossed to itself, then two flies which are heterozygous would then produce, on average, 1/4 of their offspring of the F2 generation being homozygous dominant, half being heterozygous, and 1/4 being homozygous recessive. If sepia is a dominant allele, then 3/4 of the population would then have the trait of sepia eyes, and 1/4, the homozygous recessive individuals, would have the trait of brick red eyes, the wild-type condition. If, however, the wild-type allele of the sepia gene is dominant, then the F1 generation, being all heterozygotes, will possess a dominant wild-type allele, and thus all members of the F1 generation would possess brick red eyes, the wild-type trait. When the F1 generation is crossed, given that these F1 flies are heterozygous, then the F2 generation will possess 1/4 homozygous dominant, half heterozygotes, and 1/4 homozygous recessive. This would result in a 3-to-1 phenotype ratio, in which 3/4 of the population will possess the dominant wild-type allele and have brick red eyes, while only 1/4 would possess the sepia allele, which would give the dark brown eyes and be the recessive condition.